becoming smaller and smaller proportionally, and is starting to interfere with the exchanging of materials. Food, oxygen, and water will all come through the cell membrane and be used by the cell to live. At the same time, the waste products will be moving out of the cell that have already been used. Exchange takes place based on the size of the surface area, blah, 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 blah. Some boring shit. Alright. Asexual reproduction is carried out by cells for two main reasons. The first reason is faster process than sexual reproduction. Sexual reproduction is the fusion of two gametes to form a zygote. This process would require the cell to find another mate and often takes longer. Cells prefer to asexually reproduce, meaning that one cell or organism will produce two genetically identical daughter cells. This process is useful for cells because the two identical cells are able to carry out the same functions that their parent cell did. Cell division is a form of asexual reproduction and is carried out by all living cells in the world. For asexual reproduction to happen, the cells must have something called a chromosome. A chromosome is a big bundle of information and DNA that is stored inside of a cell. A chromosome is extremely important in the process of cell division. Without them, there'd be no way that cells could asexually reproduce, and the results would be disastrous. If we were to look inside of a eukaryotic cell, they would have more chromosomes than any other cells. This leads into the actual process of the cycle, the process of mitosis and cell division. The key to why things are able to live the way that they do. The cell cycle is actually defined as the process where a cell grows and pairs with division, and then divides into two daughter cells. Each daughter cell will then move through the cycle again, and it repeats itself. The cell cycle consists of 5 main phases that we know in research. The first 3 phases are in the category of the process of interphase, or cell growth. The G1 phase standing for gap 1, is where a cell will be growing and actually carrying out its normal function.
You're all doing. Why the fuck did we tell us today?
It is classified as growth and development stage. The S phase, synthesis phase, is where DNA, the chromosomes, will replicate inside of the cell. By the end of the S phase, the cell will be filled with 2 copies of DNA. Finally, the G2 phase standing for gap 2 phase, where the cell will get ready for mitosis. Where the cell does many functions, and it is the last point in time when the cell will be one whole. The next 2 phases are under the process of cell division. The first is called mitosis. This is where chromosomes and DNA will separate, and the cell will have 2 exact copies on each side of it. The next is cytokinesis, is the division of the cell membrane. This is the last phase of the cell cycle, where the cell actually splits into 2 different identical copies. At the end of this cycle, the 2 daughter cells go back into the G1 phase to grow. There the cycle begins, the process is all over again. There are 4 phases that make up the process of mitosis and cell division. Mitosis goes in order of prophase, metaphase, anaphase, and telophase. In prophase, the chromosomes will start to gather on the nucleus. The chromosomes of the nucleus will start to gather inside and duplicate together. They all bunch up in the nucleus and create chromatids, which each are a strand of DNA that bunch together. These strands are attached by centromere that form into a sort of X shape. Centrioles in the cell will produce spindle fibrils for later on. Metaphase in the process is the point where the nucleus has broken down. It is where the chromatids will line up along the middle of the cell and have a spindle attached to each center of the X shape on the centromere. During anaphase, the spindle will split the chromosome and 1 copy will go to each side. They are pulled by the spindle when the side attached to the centriole starts to disintegrate. The last and final phase of mitosis is telophase. During telophase, the nucleus will start to reform around the DNA that is going to each side, forming 2 different nuclei. At the same time, the cell will start to pinch in toward the middle, leaving 2 exact copies on each side. After the entire process of mitosis ends with the splitting of the membrane, after the entire process of mitosis, it ends with the splitting of the membrane, which, if we go back, is called cytokinesis. The thing is that animal cells and plant cells use cytokinesis differently. In animal cells, the cell membrane will pinch and split into 2 different cells. With plant cells, they do not have a flexible membrane, therefore there is no way that the cell can split. Instead of something called a cell Instead, something called a cell plate forms inside of them, splitting the cell into two different sections. The last thing that comes into play is called cyclin, a protein that speeds up the cycle. The problem with it is it can create cancerous tumors that are able to completely kill humans. This, however, is useful because it tells the cell when to divide and is known as a division factor.
Is that it?
That's it. That was the introduction.
How many pages was it?
4.
Only 4?
No. Yeah, 4.